Hello everyone, my name is Alexander Campos and I'm very pleased to be joining Donald and Anya today for a discussion. Just to give you some information about the panel today, or actually the artist talk discussion presentation. It's a mixture of soil and art and community projects. And we'll tell you all about the details of the projects, but just give you some information about the speakers. Dr. Anya Paltseva is a native Russian. Anya holds a PhD in Earth and Environmental Science from CUNY Graduate Center. In addition to her academic exploration, she's a research and program coordinator at New York City's Urban Soils Initiative. Her experience is assessment of bioavailability of heavy metals in urban soils. Anya has presented her research at national and international soil conferences in Italy, Mexico, Brazil, China, Russia, and authored and co-authored 17 manuscripts and book chapters, with more on the way. And she also teaches as a speaker and lecturer at Brooklyn College and New York University. Joining her is Donald Daedalus, who is an artist living and working in the largest city in America, called New York City. He was born and raised, this is Donald, as you'll get to know him. Yeah. He was born and raised in the shadows of the nation's largest public observatory, educated at the University of Washington and San Francisco Art Institute. Through film, photography, sculpture, and installation, Donald works draws attention to idiosyncratic and nuanced contradictions and assumed truth. Since 2006, the Ziluma River Project, which we will be discussing today, has followed the national concern from invasive species into the more complex debate of biodiversity and existential issues of anthropocentric epoch. His work has been shown around the city and around the country as well, including SPAC Mountain here in the city, London, White Lady, and so forth. So we're here to them, so I'm going to stop here and ask them to talk about their projects. I think Anya's going first. And there's a brochure upstairs. There's a next small exhibition upstairs that you're more than welcome to see if you haven't seen it already afterwards. And I have to say, the presentation is being filmed, so there we go. And if you're going to ask questions after the presentations, we ask you to use the mic up front because Donald is recording it.
I'm very happy to have you and to share my research with you. I'm a soil scientist and I study urban soil contamination. And next, so you can see. Next. Around the United States and Canada, there are about 18,000 community gardens. And just in New York, there are about 1,500, highlighting the importance of urban gardening and engagement with soils. The community gardens and home gardens bring lots of benefits to people, of course. It's nutritious food, it's community building, it even reduces crime activities because people have something to do. But it associated with associated with some hazards. What are the hazards? You probably heard. It's lead and commonly arsenic that are found in urban soils. And the problem is them been for even thousands of years, but people still using and still put them into the soils. Lead was discovered like 5,000 years ago. Ancient Romans would put lead into their wine to make it sweeter. We put it in gasoline, and then we breathe it in, we put it in the soil, we eat vegetables grown in those soils. Arsenic came at a later time, but commonly was used as pesticides. Next, please. And the main sources of arsenic are the pesticides, lead arsenate, that was used against gypsy moths and was sprayed in northeast of coast of the United States in orchards. But it doesn't go anywhere, it stays in the soil. And lead, it's commonly from paint chips from the old houses, soils, point source emitters, and some gasoline. Next, please. Why are we concerned about those metals? Well, lead is neurotoxin. It affects cognitive development, especially of children. They are acute drops with the increase of blood lead level. And arsenic is carcinogen. It can cause some cancer. That's why it's important to study those metals and see how they can be uptaken by plants or by humans. Here you can see the current status of lead in New York City garden soils, and the majority or the highest concentrations of it's not working, but the highest concentrations of lead were found in Greenpoint, Williamsburg, downtown Brooklyn, Prospect Park. Those areas you can see in red on the slide, and it's very. I started hearing the noise. Someone lives there. So you should test your soil, and we can talk where you test it. It's right behind this wall. We have the soil lab at part of New York City Urban Earth Institute. You're welcome to bring your soil here or send it here, and we'll test for you. It's really important to know, and you don't want to grow vegetables, especially if you have kids, for kids to play in that soil. Other areas are more safer, as you can see, like in yellow, and typically concentration drops from the inner city toward the suburbs, because in the inner city it's where most of the historical contamination was from industrial sites. Next, please. And here, another one. On this map, you can actually see a subset of 126 gardens that I chose and overlapped with some historical sites in the blue and some current sources of contamination in yellow. Those are buffers, and some of them, like in brown, they were overlap. So a lot of gardens are contaminated from past industrial use, like legacy lead, manufactured smelters, coal yards that existed between 1764 and 2004. Those considered historical contaminants. Some of the manufacturers may potentially release now contaminants as well, or some waste transfer stations or major roads that are current sources. Next, please. But it's important to know is that it's not the total concentration that is harmful. It's bioavailable. What does it mean? Those maps were constructed using total concentrations, and it's what mostly you're going to test when you send your soils to the lab. It's important to know how much can be uptaken by humans, and not all lead is bioavailable, meaning it's not all harmful, but only fraction. And to know that fraction is actually very difficult, and it's what scientists have been working for decades, and it's what my PhD was about. So bioavailable is how much lead can get in, or any other contaminants, get into human and affect tissues and organs. But it's time-consuming to do those analyses because they're tested on animals, and it's unethical and very expensive and long. So scientists try to find other ways how to measure it in the lab conditions, and that fraction would be called bioaccessible. The idea is the same, but just different measurement techniques. Next, please. And I did this bioaccessibility studies, about 50 samples in New York City, to look at the variability. And what is interesting, we don't see the correlation between total lead, you can see on the graph, and bioaccessible lead. There is no correlation between the two. And we measured the two different pH ranges. That is the protocols that are used by EPA. 1.5 is the pH of our gastric solution, and 2.5 is our proposed method to measure it. So in both cases, we don't see any correlations, highlighting the importance of measuring bioaccessible lead, or bioavailable, but it's not available for public testing, to actually know how much is harmful. But we're able to use some equations to convert bioaccessible into bioavailable, and I came up with this table. Out of these 49 samples, 70% were actually at low risk, which is a good thing. Couple samples were like medium risk, and 27% were at high risk. So most of the samples were still at low risk, which is good. Even at the high concentration, as you can see in the graph, at the high total concentration, we may get very low bioaccessibility, which means we need to measure bioaccessibility rather than total concentrations. Here's a case study from suburban New Jersey. We did some studies in a suburban farm in New Jersey, and they have a contamination with lead and arsenic from a lead arsenate pesticide used about 100 years ago. It doesn't go anywhere, it stays in the soil. So they invited Brooklyn College to do some analysis. We amended the soils with different treatments. We used bone meal, triple superphosphate, manure compost, and raised bed compost to mitigate that soil and arsenic. Why lead and arsenic? Why those amendments? Bone meal and triple superphosphate contain phosphorus, and when phosphorus combined with lead, it forms paramorphic mineral, which is very stable and not bioavailable. It's what we want to have. And compost have the same target. They form organomineral complexes that are also unavailable for humans, and they are, even if you ingest this type of soil, it's not going to harm your organs, it just goes through the system. And it also dilutes the soils and bring the total concentration down. So what we found here that some vegetables like carrots would take more lead and radishes would take more arsenic. Tomatoes were the safest vegetables to produce, even in contaminated soil. And lettuce would be contaminated through the splashes and dust from surrounding areas. So it's really important to peel your produce, to wash your produce, but for carrots it will not help because the carrot's contaminated through inside. You also core inside of the carrot, that's the mechanism, it's through its uptake, but it will help for other vegetables. So choose fruits over roots when you can plant in contaminated soil. You can still plant in contaminated soil, but just need to be careful what is the level and what you can plant. And there are some guidelines at which level you can still plant different vegetables, so lead and arsenic. There's also paper on the table from this study. You can read in more detail and some recommendations that I provide. So where contamination coming from, it's some of it can be uptaken through the soil by roots. Some of them it's just splashes and dust from surrounding areas. But the taller the plant, the safer typically that is. It has very strong physiological barriers inside, and it's further from the soil. We found among these amendments that compost was the most useful, and plant type is more important than amendment type. Different plants react differently on amendments. So it's a little bit tricky with amendments in urban areas because the soils are very heterogeneous. They're very variable here. Next, please. Another site in Brooklyn, we wanted to see what is actually main impact of on human. Is it plant uptake, like vegetable contamination, or it's a soil dust ingestion, or it's dust inhalation? We found it's actually through soil ingestion. And it's mostly harmful for kids. We use the little kids, less than 6 years old. They're typically most susceptible organisms, and we found that next you can see the guideline. FDA guideline is 3 microgram per day of lead, but through soil ingestion you can get up to 17 or 26. We also had different amendments there, so it's the main path, and only after that it's followed by vegetables and dust. So it's really, you want to have 80 to 90 milligram per kilogram of lead in the soil to be safe, although current EPA standard is 400. But to make sure the kids are safe, less than 100 should be adapted. So what is the best mitigation strategy? Currently, we think it's using, bringing new soil, like a clean soil on top, building a raised bed on top of the contaminated soil, and mixing it with the compost. Some of you maybe heard, and you can Google and look for Clean Soil Bank. It's a program that's been developed by Mayor's Office of Environmental Remediation. They propose to use glacier sediment that is clean and excavated from New York City before building construction. So that sediment can be mixed with the compost and use it. Or you can try and find a way how to remediate with amendments, but it can be a little bit tricky. Need some research to be done to be effective. Next. And on this note, I would like to thank you. And you can ask me questions after Donald's presentation.
Thanks, Mary and Margaret, for inviting me to come out here and coordinating this. Is a really fun installation because it's a bit between camping because of the ferry schedule and electricity and Wi-Fi, and there's no toilets. And then also like... Or running water, yeah. But there's a lot of passion, and somehow it all got done, even the projector. So thanks so much for having me out here. So, and Anya, thank you for all your hard work and collaboration, etc. I'm going to talk about this project that I started in 2006, but I didn't really know that I was starting it at that time, and it's an umbrella project that includes a series of artworks that I've made, but in retrospect, I've divided it into these 3 stages. Acquisition, conversion, and distribution. There's a 4th stage that's not quite there yet, but these 3 stages are there. So could you go to the next slide? So how I imagined this project was utilizing art as a platform for funding, advertising, and basically combining things that I was researching and visualizing that, and then implementing that for this problem of invasive carp, which is like this long, strange passion I've had since 2006, and using the carp as like a fertilizer or resource initially. So that's kind of stage 1, 2, and 3. This is an improvisational chart, but we can go to the next, and we'll dive into some of this in detail. So acquisition. So yeah, in 2016 or 2006, somehow I ran across this. I think it was on YouTube, but it doesn't even sound right because were people watching YouTube in 2006? Yeah, I mean, I might have seen it on television at that. I might have had a television at that time, but I saw this video. If you can go to this video, it's stuck in my mind. It's not this exact video. I couldn't find the actual one, but if you go to the next slide, if you would play this.
Easiest fishing ever, huh?
So the video that I saw was basically this video.
Yeah, no, they're slimy too.
And yeah, I'd never seen anything like that. And since I've seen it in real life, I've never seen anything like this before. It's almost biblical, actually. It's like something falling from the sky or just exploding out of the water. If you can go to the next slide. So around 2013 and 14, I began thinking about how I could actually go and catch this fish, and I've been following the evolution of this problem. In 2006 is when it kind of got to a national level in the form of an event called the Redneck Fishing Festival or Tournament, and it's in Illinois, in Bath, Illinois, and basically a very small town started this tournament to go and catch these fish as an effort to actually deal with the invasive population. And I'd always wanted to go, but I'd always been so broke that I couldn't actually get the resources to go to the middle of the United States. But I began thinking about if I could go, what kind of thing would I need in order to catch these fish? So I designed these apparatus that you can connect to a sort of a conventional boat. This is the first prototype of acquisition. If you go to the next slide, there's some more detailed shots. But basically, it has some arms with like nails to catch the jumping fish. And one thing I should mention as I see your facial expressions turning a little bit, with invasive species, at least this one, and don't quote me for every invasive species, there's no limit to like how many you can catch, and even the conventions of how you catch them. So I know to like people in the greater metropolitan area, this boat might look a little bit medieval, but it's actually remarkably similar to what the U.S. Department of Fish and Wildlife used to catch the fish, which were boats that had electrical fences above the water, so when the fish jump out, it would basically electrocute them to death, and then their bodies would just float downstream, probably to St. Louis or somewhere there. So yeah, there's a facial expression. Like that's taxpayer money. And then this boat basically had this aluminum wire net at the front that was supposed to catch the fish that were jumping in. So this is like thinking like the characteristics of this fish, how could I address the fact that it jumps from the water. It's about 6 to 10 pounds and about 24 inches in length. And it's known for hurting recreational boaters. Maybe I should backtrack 1 second about this invasive species. Yeah. Yeah, so one of the people in this town that organized the event described it, it's the equivalent to being hit in the face with a bowling ball. And he'd been working as an emergency room individual, I don't know, in this small town. They had kind of any role that you can fill, you can take it. Yeah, so people will be driving down the water, and essentially the motor boat scares this fish, and it jumps out of the water, and it hits people's face. And so, for people who don't live on the water or the river, they're kind of like, just don't drive your boat very fast, right? That's the solution. But the history of the fish is something like they were introduced for wastewater treatment plants as a method to control the level of phytoplankton. And basically, the treatment plants flooded, and the fish, which were primarily like in the southern United States, have now spread to almost all but 14 states in the United States. And they're nearing to the Great Lakes. And essentially, they're the most successful aquatic organism in North America, and native species that either represent biodiversity or something that people want to fish or eat. Their populations have been declining. So the danger point is when these fish get into the Great Lakes, they're going to decimate obviously all of that fishing industry and jobs and nutrition that comes out of that, but also then they're going to have access to Canada. So the U.S. Fish and Wildlife and the Canadian government have been spending billions constructing various ways to stop them, which are like dams or, locks or, all kinds. There's literally there's a new method of trying to take care of this fish every year that comes out. And it's interesting because people in New York are like, oh, we don't even have a problem with this. But actually they are in New York State, but they're just not in New York City yet. So we can go to the next slide. I'll try to speed this up a little bit. So there was no practicality of actually taking this boat from New York to Bath, Illinois, so I tried to take some of its components and shrink it into something that's wearable. So this is my kind of like Power Ranger suit for competing. And we could go to the next slide. The tournament is set up in heats. Each heat is 2 hours. There's 2 heats per day. There's 4 heats. There's 2 days. They take out about 10,000 fish in 4 days, but even with that, it's only happening once a year, and the population's still growing. In my boat, we caught 52 fish, and we lost. The number one boat caught 272 fish in 2 hours. And they basically had a volleyball net in the middle of their boat, and the fish just jump into it. And you could see this is about a ton of dead fish. You could hit play on this. And they're ultimately loaded into this refrigerator truck and driven about 100 meters from Thompson Federal Penitentiary, which was where Obama wanted to move all of the prisoners from Guantanamo Bay. And so there's all those kind of federal issues going on in the middle of Illinois, and then they're turned into a fertilizer. But the fish have a strange capacity when they jump out of the water. Their gills, because they're optimized for breathing in low water, they overexpand with oxygen and they explode with blood. So every fish, when it jumps out of the water, is just covered in muddy, toxic water and fish blood. It's a crazy event if you ever want to go to to Bath, Illinois. It got a lot of exposure on like BBC and I think ESPN went there when I was there. There was a gentleman making a documentary from National Geographic. So we can go to the next slide. So the idea of actually like using these fish as here's another characteristic of this fish. I can't believe I left this out. There are two reasons why people don't want to eat this fish. One is that the American palate doesn't really eat very much carp. We don't really want much carp. And the fish is about 70% fish bone, which means that it's not commercially viable to hire people to fish and pay them by the pound for the fish, especially for meat that nobody wants. So the characteristic that it's 70% bone was beneficial to me because I had learned that there's a compound called apatite inside the fish bone that bonds with lead, and it forms pyrophosphate, as Anya was saying, which makes lead in the soil no longer bioavailable. And so the idea was that I catch this invasive species, process it, and then use its fish bone to lower heavy metal pollution in various places, mostly around post-industrial areas in the U.S. But just participating in the redneck fishing tournament wouldn't be enough because it's only 10,000 fish, and I wanted to actually have this functioning somewhat like a business. And I began researching how other people catch fish rather than sending them out on a boat. And this is like a traditional Chinese dip net. Basically, it's along the shore, and you have a cantilever, and you weight it, and you push into the water, and you put some kind of a bait. The fish go, and you pull up, and you take the fish out. And since it's an invasive species, you could then catch the fish in ways that are not legal for commercial fishing, but you could commercialize it. So we go to the next slide. I traveled to Vietnam to learn how to make this. The structure it's Chinese dip net, but it's also used in most of Southeast Asia, called the vo bay here. And this is in Cu Chi, Vietnam. And we built it. It's really rudimentary, but it's kind of brilliant. It's so simple, really inexpensive, mostly bamboo. The net's the most expensive part. We can go to the next. So that's the third prototype. So after I caught these fish, it was like the question, like, okay, you've got these. I decided I would bring 5 back to New York to test how to process them if I was making like a mock version of this workflow or business model. So I vacuum sealed them and I put them in my luggage and brought them. It was my freezer for a few years, these big fish, but this is the next step in how to do this. So I realized that you can't allow dead fish to just decompose at its normal rate because it stinks too much, and it's not practical for doing it for any kind of decomposition. So I began looking at ways to rapidly decompose organic matter, and I learned about this black soldier fly. So this is the first prototype of trying to create cultivate this fly. You could go to the next slide. And you could hit play. So if you guys are composting, you've probably seen this before. These larvae, they're totally innocuous. They have a pretty short lifespan, non-vector fly. They live about 6 weeks. You can go to the next. And this is what they look like right before they go into pupate. Hit play. They're really kind of amazing. They're like the most curious little insect. Okay, so that was like the stage to try to just understand like basic situations. And what I learned in that situation is like you have to kind of mimic springtime cycles, and you have to have daylight and a certain amount of temperature, certain humidity, temperature, things like that. And this is the second creation of that, where I've added an aquarium to create a hydroponic system to grow these plants, which are necessary for the adults to mate on before they lay their eggs, which I then used to decompose the fish. And the larvae basically can eat incredible amounts, as you'll see, in a very short amount of time. We could go to the next slide. This is an attempt to, you could see them moving over a time lapse. I was trying to mimic the next video. Oh yeah, here we go. So this is a Swiss group that's also looking at the use of black soldier flies. This was in a 12-hour period, and there's two, I think they're 4-pound trout that are completely devoured. There's some caveats to this. The larvae really only eat during a certain period in their development, and then they go into pupate, and they basically don't eat at all. But you can see this clock running. You could see this video on YouTube also. It's kind of inspiring if you're dealing with organic waste. They're also, black soldier flies are used in agriculture, agricultural waste management. They're also grown for feeding to turtles and chickens as a form of protein. And that's it. It's like 11 hours and you're just down to fishbone. So that was the goal. Like, how can I create a situation like this? And subsequently actually found all of the details of how to create this exact situation. But basically you need the larvae during about 6 days to 12 days old, and they'll eat this quickly. Okay, can you go to the next slide, actually? So I decomposed the fish bones to, this is 2 of the fish, and then I dried them out, and you could go to the next slide. Put them in the oven. Next slide. And this is and then ground them up, and you've got this is the commercial fish bone, I guess. There is a company that makes fishbone meal, but I don't think anybody uses from invasive fish yet. So you can go, each fish is about 1 cup of fishbone. And then I had the idea, okay, well, I should go out and put this in some soil and see what it's like. And then I realized that, you go to the next slide, that there's not a lot of dirt in New York City, like a place to just access dirt or even test dirt. I mean, there's gardens and things like that, but pretty much so much of the surface is covered. So I used a map from a project called Ghost Factories by USA Today, and they're basically mapping all of the closed factories that had some kind of a metal smelting activity, and you can check it out online. It's pretty amazing. I think all of your research will probably point to the same thing of like North Brooklyn, Williamsburg. But I had found this area in the South Bronx, I live in the Bronx, where there was a factory that was smelting lead, and there was like a patch of open land next to it. So I took a soil sample there, and then on the other side there was a small park. And I thought, oh, let's just do this as like a control. And then I did it in another park where I live. If we go to the next slide. So these are the 3 samples which I sent to Anya's lab, and she tested them. And if you go to the next slide. So this is the park that's adjacent, and then the factory's over here. And next slide. What I found was kind of surprising. Actually, the playground had higher amounts of lead than where the factory had been. Now, I don't know like what has actually transpired since that factory went out of business. But if you look, there's an industrial index in the Science Engineering Business Library, SIBL on Fifth Avenue, and they'll list all of the companies of an industry, and you can see their address and when they opened and when they closed. I think this one closed in the 50s. But what I did in this picture, aside from overlaying it with some of, just analysis, not necessarily bioavailability, but in the background, you can see there's a public housing project, and then I collaged in about 14 images that are time-lapsed over this and put in the cars in the background, which don't look unusual, but there is something questionable about having like a playground adjacent to, like the Triborough Bridge, basically going onto the Triborough Bridge. And there's nothing unusual about this photo. I mean, you're just looking at it, you're like, oh yeah, it's like a park, a playground. But my hypothesis about how this lead got there was from leaded tetraethyl lead gasoline and particulate accumulation that was phased out in 1996 from the Clean Air Act. If you go to the next site. So I went to that park and I integrated these fish bones. I basically had 2 different fish, how I'd slightly dried it differently and tried plotting out like a control, whatever, putting little stakes in. But after I did it, the next week, somebody had removed all my stakes and it snowed. So, I don't need to test the actual conclusions of it. But essentially, a small part of that land has some dead fish in it. And then the third, which is, or the second way of doing this, dealing with this fish, I mean, it really just became like, how do I even find a place to put fish bone without having any authority to do that, like no jurisdiction, no permits or anything like that. So I don't think you can see it in this one, but I could just describe it. Basically, I found this like median in the middle of a road, and there was a little garden. I went and buried another fish there. So that's it. Thanks for your time.
One is I'm really interested in bioavailability versus bioaccessibility. And my first thought was like, okay, obviously something changes when you ingest the lead, or when an animal ingests the lead. It's exposed to enzymes and acidic solutions, as you said, in your body, and that's when it undergoes a change that makes it. Is that correct? Okay. So when you were testing it outside of an animal in vitro, you said, were you just exposing it to an acidic solution, or were you actually extracting enzymes from the bodies of animals and like exposing it to that?
Yeah, that's a super good question. I didn't go into detail, but yes, we're actually making a solution, separate solution. There's an EPA protocol that is using glycine. Glycine is known to mimic our gastric system the best as it can be. And the idea is to use the solution outside of actually the stomach and using different acids. So we use glycine, and we add HCl, hydrochloric acid, to make it acidic. Our gastric system is typically between pH 1 and 4. So at 1.5 is the typical pH that EPA measures it. So it's like the most conservative, as much as it can be digested or uptaken at this level. So we had to recreate this in the tubes, basically making this solution outside of the human body, and then we would mix it up with the soil, extract it for like 1 hour in a particular temperature, and then we would analyze it in the mass spectrometers.
Okay.
Yeah.
And then for tomatoes, are they only good for growing in lead-contaminated soil, or any kind of? Are they like resistant to uptake of any kind of contaminated soil?
For the most, I tested for lead and arsenic, but it will be good for most of the metals because not just tomatoes, but any fruit, vegetables that fruit vegetables that are tall, they have physiological barriers inside of them. So the first physiological barrier is between roots and stem, and the second one between like stems and leaves or a fruit. So the plant itself doesn't want contaminants go there. So it does not have much of uptake. But of course, sometimes like it's a crazy amount of contaminant, it may go through the barriers as well. But it's typically like, I don't know, you have to grow it in a mining area somewhere. In an urban environment, it should be safe. I grew some tomatoes even at like 2,000 parts per meter, like 5 times more than EPA, and tomatoes were still fine. So it's really important to grow something tall for these physiological barriers, or because it may get splashes from surrounding areas like lettuce or herbs. So growing tomatoes or like eggplants, even cabbages, because you only eat inside part, or any like squash that is growing up, you'll be okay.
Okay. Thank you.
Thank you.
Well, we have some more questions, but we have questions that I'm going to help you to ask some questions as well. And because we have a limited time as well, I think it's already 2 o'clock and there's the next lecture going on. So before we ask, I want to ask, can you talk more about the collaboration that you really need to share that? And of course, that's a very also parallel, issues in a certain way, and the carp and the soil relationship. I also want to talk about some of the community engagement projects you've done to really bring
attention to the issue of carp and soil.
So yeah, I can say how we started collaborating. I had wanted to take my research to the next level, so I took some classwork at the Botanical Garden, and coincidentally, I mean, this was specifically for this project to learn about soil chemistry, and there were 2 classes, and I had to sign up for one of them, and then I was like, should I pick this person or this person? And I pick Anya. And and the first day she told me about the research, I said, this is perfect. So that's how it started.
Yeah, it was like about 4 years ago, and since then we just started to, talk. And because someone needs to make those amendments for me to work with them, right? And it's a great way to create those amendments, because you're not just use some kind of fish, but you actually use invasive species and improve the ecology of the river and stuff. So I think it's a great way, and if it can commercialize it at some point, it will be nice. And in some areas, like in California, when they had a lot of fishery, they use a lot of fish bones, and it doesn't always smell great when they amended the soil, but it does work. There's some successful stories. I haven't used specifically fish bone in my experiments. We used commercial fertilizers that any gardener would do. Like I just tried to mimic what any person would buy. We went just Home Depot and bought whatever was on the shelf. And so.
Did you want to talk more about how you tried to use it more from the artistic side, bringing more attention to it?
Are you talking about as a platform for invasive species? Or
well, just how you're trying to kind of create jobs or the emphasis of using it as an economic resource while taking care of the carp.
Yeah, so the 4th stage was to use fishing as kind of, it's not really shared economy, but almost a gig economy. Like anybody who wanted to could fish this boat, could fish this fish. And particularly trying to coordinate with people who are recently out of prison and potentially black boxed, but that stage hasn't been implemented yet, so I didn't really want to talk about it a whole lot. But that was the idea was that this is such an abundant problem, but also it's a problem that can be addressed with people who don't necessarily have to have a very sophisticated skill set, or even have to have certain hours that they want to work, or even work a specific duration. If you just wanted to go out and catch invasive carp, you can just do it Friday, Saturday, and Sunday, and then do something else the rest of the week, or certain days of the week. And also you wouldn't have to have a fishing license and something like that. So that's the 4th stage that's supposed to have this whole workflow going, and if you want to support it, there's a donation box upstairs in the room. You want to? Yeah, go ahead.
Yes, I think somebody had a question.
Yeah, it's taped on there. It's taped, yeah. All right.
So, some people look at invasive species, not just animals, but plants, from the point of view that there is not such a thing as an invasive species, but an environment that is out of balance, and I like that idea. So, taking it from there, my question is, in places where this type of fish is more native, what does that environment look like so carps don't take over? Because I think that's, I like that approach, so yeah, I'd like to hear what that's.
Yeah. So coincidentally, I was in Austria, Central Europe, in November and December, and they are native there, and I had the opportunity to speak with a New Jersey marine biologist who had been stranded or living in Central Europe for several decades. And some things that he talked about with that fish, first of all, they have traditional carp for Christmas. I guess you could, like in Prague, you can find these carp where they're alive, and there's, I guess, some traditions where people keep them in their bathtub until the day of Christmas, and then they kill it. Have you heard of this? Okay.
We don't use it in Russia.
In terms of, yeah, I did ask him about some characteristics of why the carp population wasn't exploding in the same way it was here. And I think it had something to do with water temperature. So, like in Austria, there's tons and tons of rivers. So if you have an aquatic pest, it can potentially be anywhere, right? And I think that particularly in the southern United States, that carp are able to live and breed nonstop, year-round, and then they don't have natural predators in the same way that they do in their native, different carp. There's 4 invasive carp species in the U.S., but some of those are from Asia, China, and some of them, I think they're collectively called Asian carp, but some of those same species are found in Europe. I think, yeah, there's some kind of social things that have evolved with carp when they're native or non-invasive, such as people eat them, either traditionally or daily. And then there's also predation that doesn't really occur here. So I think I think you've got a really good point, though, about how we have conceptualized a life form as a problem. And I think I guess if I were to defend the implied scientific perspective, it's that invasive species problematize our potential to predict things by disrupting what's perceived as a stabilized environment. In the same way that you can say that some kind of climactic change is a problem because it problematizes our capacity to predict the future, whether that's growing, food or something like that. Did I answer something of your question?
Yeah. That makes sense. But then again, if you look at it from a historical point of view, environments evolve. Like our environment is not the same now as 2,000 years ago.
Sure.
So I guess it doesn't make sense to want to keep everything as it is right now or as we like it. So things evolve.
Sure.
Yeah, and I think that that's a really good point. There's the original. I think what you're describing is like the originalist versus non-originalist perspective on ecology, and it's maybe most easy to compare invasive with non-invasive or native, but you could also think about it as how I've seen this issue evolve was originally from invasive to non-invasive, and I personally looked at it and I think, oh, that's strange, like the vernacular is very similar to how we talk about people who are native or non-native, right? But I think this problem has also evolved, how it's conceptualized by the public, particularly in light of like the United Nations report that came out 2 weeks ago that talked about like 1 million species that are now on the extinction list. So if you think about native not in comparison, or non-invasive not in comparison with native, but on a continuum of native, invasive, native are sort of like average at surviving in an environment. Non-native are problematic because they're too good at surviving in an environment. Extinct are problematic because they're not good at surviving in an environment. So if you want to address it within that trifecta, it's like, do we want to preserve native? Do we want to preserve those that are about to become extinct? Do we want to discourage or dissuade non-native? Or do we could also just say like, hey, we can think about like, we've got all these fish that will reproduce. Let's just everybody start eating carp, right? That's totally another way. Yeah.
I had a comment in regards to this. I think we can all agree that this is an environment that's out of whack. right? And it's not good to have too much of one type of species because it's not enough biodiversity. So a point that I feel like we didn't discuss enough is the water temperature thing that you were talking about, because what you were saying is that there's ways to adjust the environment to help put it back into whack without necessarily having to remove the invasive species, and that's done in other areas by introducing a predator or whatever it may be. But water temperature is something that we can't change, right? So we need to apply other solutions in the situation that maybe you wouldn't apply in another situation in regard to invasive species. So that's just something that stuck out at me that I wanted to highlight.
Any other witness questions? So I think well, I just want to ask you, can you talk a little bit about the Institute and how you are reaching out to share the information how people could actually take advantage of the lab here, get more involved in caretaking for their own soil, their own little land, their little box, whatever they grow vegetables in.
Yeah, so New York City Urban Institute, it's a platform for everything related to the soils. And we are institute of full of collaborators, not just in New York, but outside of New York City boundaries and even abroad. So if you have something that you would like to share, to expose or organize, or we are the platform to talk about it. If you want to just to test your soils, we have a lab which is actually opening up this, I don't know, like a week or two ago here on Governors Island, actually literally crossed behind this wall. We have a second location in Floyd Benefield in Brooklyn. So if you go to our website, you can just Google New York City Urban Soil Institute, you will see test your soil, and then you'll send your sample and it will be analyzed, or you can drop in both locations. So if you're here, you will also see the price list of what you want to test for, and you can test for nutrients, nitrogen, potassium, phosphorus, for organic matter, for pH, for salts, and for heavy metals. You can get total concentrations. Bioaccessible concentrations are not easy to measure, even with this in vitro method. And right now, it's available in a couple of places, and I think it's Ohio State University for sure, maybe Kansas as well. I do it only for research purposes, not for public. So if you want to send it, it's very easy, and then you'll get results in a couple of weeks by email. You'll get the report, and if you have any questions, you can always contact us. The director of the Urban Soil Institute is another across the hallway, Tatiana Morin, blonde woman, so you can talk to her to know more. And we have the whole, not the whole, at least part of the building is collaborators of the Institute, and Donald and I have an exhibition upstairs, so stop by and you can see more photos of our work and some lead sources and some amendments, and you can talk to us if you have more questions.
I want to thank Anya and Dama, and of course all of you for coming here today, and of course the Institute and Mary and Margaret, who have, there are two artists who have a grant or lease on the building for 3 years, who invited all these, urban, social, soil, land projects to be here together. So they have a residency program too, where they invite artists to have studios as well. So please look around, enjoy, and thank you. Yeah, what's yours? Because I need to set up the next lecture.